We are so excited to have Saad and Vishal here from Georgia Tech and UCSD to tell us a bit about how they got tangled with one another in some research.
So yeah, we're going to be talking a little bit about how we started working together, and how we fell in love with worms and tangles. This collaboration has been a really big part of my career as well up to now. So I'll say when we started working together, I was a grad student, and it's been a few career transitions since. And now I'm an assistant professor at UCSD, and we're still collaborating. So hopefully this will continue for some time.
And I'm Saad Bhamla. I'm an associate professor at Georgia Tech. On paper, I'm chemical engineering. All my degrees are in chemical engineering, but I consider myself a biophysicist, and hopefully you'll see some evidence of that. And in today's talk, we were kind of inspired by Anne's kind of a free flow jazz spirit, and we're going to try to do that ourselves and tell you a little bit of story of the science, but also kind of behind the scenes of how that happened. And so the primary kind of anchor for us over here is this paper we published in 2023. At the bottom, you see Vishal and Harry Tuizon. I don't know if the mouse pointer works, but they were co-first authors. Vishal was started as a PhD student at MIT and then an independent Stanford
Science Fellow, yeah.
Stanford Science Fellow. And then Harry was a PhD student in my lab, and I'll show you his picture. And that was kind of the main project, and we'll show you a little bit that's going on. And we've also written a review paper together. And Vishal will tell you a little bit about the right-hand side of this.
Yeah, and then since then, and currently we've sort of branched out into a few more current projects about knots and tangles. So in the worm space, there's many other cool behaviors they can do involving forming single knots of different types of structures and braided structures. But then this collaboration has taken us to many different systems, including cotton threads and bacterial colonies that tangle up, that we'll say a bit about towards the end as well.
So the way it all began, I pulled this email out last night, and I wrote a cold email. And in full disclosure, that's not the only cold email I wrote, but this is the cold email that got a reply. So I wrote to Jan Dunkel, and I said to him, look, I came across your fiber knot Science 2020 paper. This was also led by Vishal. They had written this beautiful paper on the topological mechanics of knots and tangles. And I said, if you'd be interested in talking about active knots, because they had talked about these, classical knots. And I said, I've studied worm blobs, and I put in a video so you can see that link. And I said, we have a paper coming out on PNAS, and we built robots on this, but if you're interested, we'd be happy to chat more. This was my 8th cold email, and I have found that...
8th cold email?
No, not to Jan. However, that does happen. I have to reach out to him quite a bit to get a reply. I say that because many times when we do kind of these interdisciplinary collaborations, and I'm still an unknown, but I was still further unknown at that time. It takes a huge leap of confidence for somebody who's at the other end of the field to say yes. And I felt I was putting myself out there, especially when I would reach out to mathematicians, people who are studying DNA knots, et cetera, who are doing theory on this, to try to engage. And it's very, very hard because kind of the reaction might be worms, like who cares? Or at least, I don't know, maybe I just go into their spam. So I share this because sometimes things work out. And in this case, it's a great story. But I also wanted to admit that many, many times you just don't get a reply, but you have to start somewhere. So what's the video actually attached as a hook? And I think this is where I had done our work. And this is Harry on the right-hand side. Harry, in a Petri dish, this is a bird's eye view on the left-hand side. So it shows real time how about 50 or 60 of these worms, Lumbriculus variegatus, tangle up. And so these are obviously active filaments, flexible, and they're tangling and braiding together to form a cohesive structure that pulls inside. But here's the kicker, right? Like that already was interesting, but the video that got us hooked with Vishal and Jan is Harry on the right-hand side brought a stimulus. So in this case, he brought a 3-volt battery connected to those wires, and he's shocking the worms. And now in slow motion, you can appreciate how the worms untangle. So really, we had the premise of what we thought was a really cool puzzle. You've got these amazing worms tangling, and then they untangle within the blink of an eye. How the heck do it do it? So that was basically the puzzle that I was proposing, that we needed their help in understanding. Biologically, it was interesting. Mathematically, I wanted their perspective and help to decipher. And so that was kind of, I thought was beautiful. What I saw over here was this beautiful biophysics problem, and I'm curious to hear what Vishal saw. Maybe either way, yeah.
Do you have a question? So can you say more about the worms? So where do they live? Like, is this a natural behavior? And
yeah, I'm going to get into it in a few slides. Yeah, this next 45 minutes is going to be like Memento. We'll put a few slides here and then, and hopefully everything coalesces together.
It's like jazz.
Yes, jazz is better.
Yeah. So I mean, when we saw these videos, I was obviously blown away. I mean, nothing does this in real life, right? And I think it's apart from the worms, nothing does this in maths, I should say, is what I. So I think the takeaway here from the kind of mathematician's viewpoint is there's all kinds of new functionalities that live in the biological world that you might not even think to consider mathematically. So I would never have sat down and said, oh, let's try and figure out an algorithm for getting 1,000 filaments to untangle, because that would have seemed absurd. But obviously it's a type of coordination that real systems do. So there's a host of new problems living in this type of behavior. And then, so this is just a spoiler for the paper. So we're going to talk a lot about the background of how we work together and how things came about. This is just to kind of show what the goal was that we eventually hit, to build something numerically that can recapitulate these behaviors. Because if you can build something, then you can explain it and understand it. So we want to get the principles out of that living system and show that we understand the principles well enough that we can design stuff that recapitulates it.
And it took a whole team. Here's Harry Tuhin, a postdoc, Emily, who was a undergrad, then lab manager, and she's now pursuing her own PhD in neuroscience. David, who's helped us with the ultrasound stuff, and Jan, of course, we needed Jan and Vishal and the worm blob. So this is just to tell you, it took like multiple universities and a whole team to make this happen. I'll back up a little bit now and answer the question of how this started. So I did my PhD at Stanford, and for the 5 years I was there, this is Lake Lagunita, and some of you who've been there will notice the Hoover Tower. And this was a time when there was a drought in California. I'd never seen the lake filled up with water. It was always dry. But I also stayed at Stanford to do my postdoc, and one day it rained, and it was amazing. Everybody, all the students, postdocs, everybody came out, like kayaks appeared, and people wanted to enjoy this water body, this ephemeral ecological niche. And I stole a pipette and then water bottle from the lab, and I took a walk around asking a simple question. Well, what kind of life emerges in this temporary niche that has been empty and dry for half a decade? And I stumbled upon where some of the parts where some of the water had receded, this clump, and I did naturally what any of us would do. I took a stick and I poked at it. And to my delight, and then my girlfriend's horror, it was alive and it moved. And I knew I had fallen in love, because I was like, oh my gosh, this is my first, I knew I was going to start my lab at Georgia Tech. I was like, I found my new research project for my first PhD student. Walking a few steps, these are not the worms always in a blob. That's where they're desiccated, but these are happy worms, and you'll see me bring out my pipette from the lab and try to suck them out, and you'll see they're Houdinis. They go back into the wet granular matter, and they kind of escape, but I managed to bring them to the lab and image them and get some preliminary data. So fast forward a couple of years, this is Yasmin, a postdoc in the lab, and during the pandemic, we started studying them in earnest. This is Lumbicorus variegatus, California blackworms. They're about 3 centimeters in length, 0.5 millimeter in diameter. So high anisotropic ratio, flexible polymers, active polymers. And while everybody in the pandemic was baking bread, Yasmin and I were playing with this living silly putty, right? This dough. This is 50,000 worms, just worms with moisture coating their bodies. And you can clearly see that the tangling provides them this cohesiveness due to entanglements. It flows at long time scales through her fingers, and she can also just drop it. It won't bounce, but it won't splatter. The entanglements kind of form this cohesive structure. Really beautiful, amazing, and Side note, I thought I would put my first PhD students, but it took me a hard time to convince anybody to work on this because PhD students would come and say, ew, yuck, and they would not work. When I showed Yasmin this, she said, I have a 3-year-old boy. I've dealt with all kinds of poop and diaper explosion. This is nothing. And she was like, I'm a mom. Let's go. So sometimes, finding the right person on the project can help. So we can make small blobs, big blobs, and write the name of the journal or your university with these worms. Of course, these are aquatic worms, and in our lab we have hundreds of thousands, if not millions, of these worms at any time cultured, and you can see they thrive in this granular environment. They clump in groups both underwater and in air. And it's just a little bit of context. First puzzle we try to solve is why these worms blob. And if you take naturally these worms out of water, we notice that they try to aggregate and search for moisture. And a clue came when Yasmin and I observed that if you put them in plastic and air, they form this hemispherical shape. And so we suspected maybe this is to reduce evaporation. And indeed, when Yasmin did many, many experiments, plotting kind of the projected blob area over time, we realized a single worm would die. But if that same worm was basically in a 1,000-worm blob, it would survive on hours. And simple math model kind of captured this picture. I won't go into more details. But essentially the picture here we had is that this is a strategy across many different worms, different types of worms, that blobbing could be a strategy to survive harsh ecological niches, such as after floods, in sewers, and my favorite video of all of this video.
Sulfur Cave is full of toxic gases. If you're going into Sulfur Cave, you can't go in there without self-contained breathing apparatus. Just like a stinky, muddy, goopy hole in the ground, and deadly to humans and most other creatures. That's full of life. My initial reaction when I first saw the worms was, wow, there's really something living in this cave. And it immediately made me think they could be a new species that probably live nowhere else on the planet. To keep these worms alive between the cave and the museum, my homemade method of oxygenating these worms is just to aerate them through this little straw.
Okay, so we have chemical engineers, we have better ways of doing this. And Harry came along, and after this BNA's paper, as we started having meetings with Jon and Vishal, started to think about how these worms are tangling. And every bi-weekly we would set up, and they would ask, well, a lot of worms is a complicated thing. What does a single worm do? And we'd sit and observe. Harry would collect videos, and we'd show them, set it up for pairwise, ternary, quaternary, and then multiples of these worms. Just starting to put together a picture of how these worms are actually interacting with each other and coming together till we come to this kind of multiple worm tangling picture, which is obviously complicated, but this is kind of a dialogue that we started in collecting data and trying to understand what's happening. I'll pause here and kind of say a few things that I think as we were getting into this conversation and this collaboration that helped set the grounds for a productive collaboration is that essentially Jon and Vishal right from the get-go got a front seat to biological discovery. We actually didn't know what we were doing. We just had kind of early videos, but as we would do these things, we would show them and we would get their input, and we would go back and collect data and try to answer, because they'd ask questions and we wouldn't know the answer, and we'd say, let's see what we can do in 2 weeks. I think this is important because it is a contrast to this typical figure 6 model approach, and I'll show you this in a second. And what I realize now in hindsight, that we've been collaborating for almost 6 years over many papers, many projects, is that we started to apply a same language. And I think all of you, if you collaborate, kind of know this. It takes a little bit of while. And I think Gabe and Maria were trying to say that maybe this whole symposium might be just to find that common language, because that takes some time. You have to nurture and protect it, and that's what we're trying to do. So going back to this figure 6, typically when I grew up in academia, this is how I was taught, and what I saw is that Raymond Goldstein has this eLife paper where he says our theoretical results are even results. And he describes this referee of a paper, because he's the editor, I think he knows. He says that the authors should let the data speak for itself and postpone their heavier theoretical analysis for a later, perhaps in the discussion. This is the figure 6 kind of thing. And he claims that many of colleagues and my own experience was the same thing, is talk about the experiments first and the theory can come later. However, he argues later, why can't we kind of do this in the biology and math community together? Why can't theoretical results simply not just be results, or could be results, and could they be integrated? And this was kind of what we wanted, and kind of hopefully we'll show you. And I think this perspective, in my mind, when I was coming up, it was like experiments and biology was the Batman, and then theory was kind of the Robin that would kind of assist. But I think we had a slightly different perspective, and our perspective was this wasn't a Batman-Robin problem, this was a Batman-Batman problem. Because I think we wanted it on equal footing right from the get-go, and we wanted to integrate our approaches right away.
Yeah, so in terms of integrating approaches and what kind of the questions are, I think when Saad and Harry were showing us all of these incredible videos, we had a bunch of very naive mathematical questions about what was going on. And talking through this was the process of speaking the same language in some sense. So I think this is a...
Just press the button, big button. Yeah, it's playing.
Oh, okay. Okay, so yeah, this video on the left is, I think, the first untangling video that Harry actually showed us. And yeah, so it raises a bunch of questions. So it's amazing, but as a bit of a skeptic, my null hypothesis is always maybe this is just random in some way. But there's a bunch of questions about the structure here. Is it topological? Well, these are all open worms, so they can technically free themselves without having to break themselves apart. So maybe it's geometrical, but it does hold together like a tangle, like you'd expect a knotted thing to do. So it's somewhere in between these two, which means that there's potential for new maths from observing these types of structures. So we didn't actually know what anything was going to be. So I was coming at this from some previous work at the time around the mechanics of elastic knots. But I think one hint that there was something maybe mathematically interesting going on here is these Reidemeister moves. So if I play this again, so again, without knowing anything really about the system at this point, one thing that you can observe here is that there are these elementary moves, which are the three types of things you can do to simplify a diagram of filaments, of a closed loop of filaments, it turns out. But the top one, R1, you see a lot of those types of loops as this thing untangles. So we didn't know if this meant anything, but it was a good hint that maybe there was something worth pursuing here, that there was some sort of underlying topological structure. I think I want to say a little bit more also about what types of, when I say potential for new math, the types of directions we were thinking here. So, yeah, so if you think about what types of interactions you can have between objects that are small but not that small, it's basically just mechanics, so contact interactions. And if they're extended and flexible, then you can have stuff where things wind around other things. And that is actually a ubiquitous type of interaction that you see in a lot of systems. So anytime you have filaments that can wrap around each other, anytime you have any type of extended object that can slightly braid around something else, you have something that is kind of in the space of interactions between topology and geometry. So seeing these worms kind of gets you thinking from a mathematical perspective that there is this space of interactions to think about. And yeah, so to sketch out theoretically what's going on then at this point, there's an idea that there are these things called topological quantities which don't depend on length, and geometric quantities which do depend on length. And somehow these worms are somewhere in between. So as an example of a simple topological quantity, if you have things touching each other, that doesn't depend on length, the notion of contact. Distance obviously does depend on length, so that's a geometric thing. But then there's this kind of fuzzy in-between thing that's relevant to real systems, which is like closeness. So in the worm's case, there's a notion of linking. So if you have closed loops that link with each other, that's topological. It doesn't depend on a notion of length. Curvature depends on a notion of length. But then there's some sort of fuzzy in-between thing, which seems to be the type of thing that's dominating all of these worm interactions. So seeing these movies and thinking about the mechanics of these structures in this way kind of starts to lead you down this road of asking, what is the actual mathematical structure here? It's something between these two established pillars, and how can we get at it? And to Saad's point about figure 6 science, just to blow up a little bit of how one way I like to think about how theory can contribute to experiments in these types of problems, because you start with a problem or experiment here, and then the goal is to use theory in a way that informs it. So the approach that we'll talk more about for this paper, and the approach that we like to use a lot here, is you want to come up with mathematical or numerical models to understand the system, and that's an iterative process of just trying to recapture it. They're almost like numerical experiments, but then the real place where I think you can move beyond this idea of figure 6 science is when there's an underlying mathematical structure or picture behind the system that maybe has new maths, but also opens up a kind of design space from which you can say this is a class of systems that we can consider. And now you can get much more control over the initial system in the first place. So this is one way to blow up that theoretical portion of this type of collaboration.
Can I ask like a very
Please.
Okay, so you kind of seem to say the underlying thing is topological. Like a kind of Platonist view would be like, oh, like the laws of mechanics are ultimately governing the atoms that constitute these worms, and so like friction is ultimately metric because like there's a notion of like, well, that's actually how things stick together is like, is some notion of this. So I'm curious, like, are we in a particular phase space of physical matter where like it's like there's an abstraction which you automatically inherit from this class of systems that like allows you to like separate those fundamentally metric quantities so as to describe them topologically?
It's a good question because it's a hard question to answer because it's somewhere in between these things. Like friction acts as an enhancer, I think. So something like, let's say me holding this water bottle is on some level topological because I have to wrap a filamentous structure around it to hold it, right? And friction helps. More friction makes this easier. But at the same level of friction, it's easier to grip it if I can grab it, the whole thing, versus if I can only hold a little bit.
So if you deleted friction, you could still, okay, if you deleted friction, you're saying there could be a topological way to hold a water bottle, but if you deleted topology, like...
I'm saying at the same amount of friction, more topological intertwining makes holding this easier. Yeah, so that's kind of the in-between picture that you have. So friction definitely important, but topology also, something like topology also plays a role. Yeah. So you want to...
I think you kind of appreciate by now we've translated from mine, hey, the worms tangle up and they untangle, cool. And Vishal is like, well, let's think about the topological structure. What's the underlying? I was like, I like that one. I like this guy. And so I think we're complementing each other and refining and sharpening the questions. And we now have established kind of what we want from this. And there's a lot of biology and behavior in here. But one thing started to become very clear as we talked to Vishal and Johan. They keep telling us we want to understand how these filaments in space are put together. And it also dawns on me at this point because colleagues at Georgia Tech work on fire ants. Other colleagues at Boulder and Harvard have been working with bees. And getting the 3D picture of each particle in space and time is very, very hard. And so we have to do it for a filamentous system. And so we tried and through every experimental trick that we knew of and method that we had in the lab to get Vishal and Jan this data, and we also wanted to know. X-ray, micro-CT, we fed the worms lead, like, oh my gosh, this was quite a long time of trying. And then this was kind of the last technique we tried, and obviously last because it worked. We figured out that ultrasound was amazing. It works. So this is ultrasound images of the worms. And in fact, this is ultrasound the same as what my wife, they use on my wife for pregnancy. You could see that ultrasound thing with the gel, the worms are in a gel. with a little bit of petroleum jelly. And it's amazing. This is first time we've applied it to worms and it works, and now colleagues have applied it to other living worm-like thingies. But essentially, it gave us a kind of this point cloud that, with some help, we were able to show for the first time, kind of peering into this tangle, the XYZ coordinate of each of these filaments, and this is about 12 to 13 worms. There are two parts here that I wanted to highlight. One, that this wouldn't have happened if we weren't constantly talking to each other very periodically and recognizing this is important. We need to keep pushing. We need this data so that it would inform. In a few slides, you'll see the topology. So, and then also we were trying all these experimental techniques. And this is where at some point I had given up. And then I had talked to many people and gotten, again, the pushback because we were trying to use worms on things that were life-saving, like ultrasound and other fancy techniques like X-rays. And one of my colleagues at Georgia Tech in the biomedical engineering said, go talk to Stas. He's an open-minded guy. So I write to him an email, again, being very, very apologetic. I'm a legit scientist. I publish in legit journals. This is not a spam email. I really want to put my worms on your ultrasound, which I know you do NIH sponsored, like amazing stuff. But let me play with your things, hundreds of $1,000 things, and maybe it'll work. And I, as you can see from this email, I'm trying to really, it's super fascinating trying to. And he says, yeah, sure. And he let us play with it. And this is Harry on May 4th, 4:47. And at 4:55 on Slack, I wanted to put this for Alice there because I heard something about dancing parrots. Because as soon as I saw this video from Harry, sometimes in life I have this, very rarely, I feel like my whole body wants to shake when I see some cool experimental data because I knew this was kind of unlocking. And the time I spent from 4:47 to 4:55 was just finding the right GIF to post for this, to express to Harry, to this first-year PhD student, how happy I am about this because sometimes in life, maybe it's when you're going camping and you wake up to the smell of bacon, or you're making grilled cheese or a tomato soup on a winter day, or you smell something, it smells right. I knew the smell right that we had kind of unlocked a big part of the story after the first untangling, that this would open up some doors.
So the ultrasound data set was definitely a huge leap in our ability to examine this structure. I would say that, to me, the problem is not still solved in the sense that it's extremely difficult to go from the thing on the left to the thing on the right. And none of the fancy image segmentation programs you might have heard of can make any headway with this problem. So to separate out, on the left-hand image, that's the raw data from the ultrasound pretty much. And one has to figure out which point belongs to which worm. So that's the segmentation problem. For this paper, the best algorithm we had was to ask Saad's then postdoc, right? Yeah. Tuhin Chakraborty at the top left there, to manually go through slice by slice and spend like a week figuring out where every point should be. Now,
to his defense, he loved it because it was a 3D puzzle. He would do it on his iPad because he would trace one worm and you have to do it in 3D, and then it would basically give one worm 2 heads or something, and he'd go back and retrace. And he does a lot of puzzles, so he thought it was the best puzzle ever to solve this. But after 3 data sets, he stopped and was like, okay, this is enough.
So it turns out this is a bit of a digression, but it turns out there's some really interesting mathematical questions here that are still open. In fact, if one wants to make progress in this direction of analyzing more of these living systems where you have these braiding, winding interactions, solving this problem is very important. So I wouldn't claim to have done this yet, but again, this was many years ago. But more recently, I've been lucky enough to work on a little bit of this problem with a student on trying to do the segmentation in some more algorithmic way. Still extremely difficult. So I say we're a long way from being able to actually take an ultrasound and push it through an algorithm and get the structure immediately. But we're getting there. And this just shows that there's many chunks to this story that were all quite difficult as well. Yeah, there's a question over there.
The possibility that you made mistake in the reconstruction.
That's a very good question. Is there a possibility that we made mistakes, Saad? What do you think?
No.
I concur. I think so. We maybe to answer that more seriously.
The noise problem, right? Because in data there might be some noise.
Yeah. So to answer this more.
Topology, that noise would make significant difference.
Absolutely, yeah. So to answer this more seriously, the types of problems you could get, I think, you follow one worm through, and then you get to some crossing, and then you accidentally choose the wrong branch. Yeah, so you have to be very careful. We ultimately had to do 5 of these. So the hope or the claim there is that if there are errors like this, they would get washed out in this larger data set. Each data set is some number of worms, around 12 worms already. So then you've segmented around 60 of them.
Also, the error chance is less because we're dealing with 12 or 13 worms. I think it grows quite large as soon as we go to 20 or 50. So we did a lot of experiments with dilating and making sure the worms were spread out, and just to make sure we got the right ones and made it safe. But there's a limit to this method. I think your intuition is right, but we found kind of the be happy work zone. Yeah. Okay, we might have to speed up a bit.
Yeah, so we'll skip some of this actual topological analysis in the interests of talking more about the story. Okay, so so after getting this ultrasound data, we're able to use some mathematical tools to get the topological structure, and then there was a question of dynamics here. So can we model these filaments using elastic equations with some randomness? This is going to be a couple of slides, but I think in real life, this was maybe 6 months of our life where we'd have these meetings every couple of weeks, and I'd show Saad and Harry a variant of something like this. So this is obviously not good. This doesn't look anything like the worms yet. And it only gets...
Our response would be, oh my God, that's amazing.
Yeah. At a certain point, I was just showing them these synchronized dances. Again, nothing like the real system. I guess maybe the thing to say here is, well, I don't know why you guys stuck around for this bit. But...
We were having a lot of fun.
I think there was a hope to get to something that looks a bit more realistic. So I think the important thing here was just to be doing something to show and then talk about experiments and iterate. But again, there was a long period of time of just that before we finally got to something that looked at least a little more plausible. I don't know if this video, yeah, a little more plausibly like one of these real worms. And it's the kind of thing that you can't really put in a paper or express in a paper very well, that process of just failing a bunch with this for 5-6 months. And in that period of time, we also weren't really making progress on the mathematical picture. We were just trying to get something halfway biologically plausible. But having got that, we then sort of turned back to looking at single worms and what they did under this escape response. So when Harry shocked them and they wanted to untangle. And this is when I think things started to open up a little bit for us. Do you want to say a few words about the...
So, we had been staring at these videos for a while because obviously we would like to figure out how the worms are untangling. So we have some skin in the game. And credit to Vishal, who observes this and starts to say, you know what, there's something about this figure 8 that might be the clue. And he keeps harping on it. And this is where I think this math-bio collaboration is so amazing because we've seen this. In fact, others have seen it. This is Charles Roos in 1999. He writes this beautiful biology/ecology paper. And he says, this is the first discovery of a worm, which is typically a benthic crawler, right? The worms don't swim. They crawl and eat debris. He says, but if you shock them or startle them, they do this helical swimming, figure 8 swimming, and move from 2D into 3D. And he's also observed the worms aggregate and blob. And he says the helical gait allows them to disperse. But even he doesn't connect this. Only discovery of a worm that does a helical gait actually holds clue to this topological puzzle. And it kind of took Vishal to appreciate that this figure 8 hold a topological kind of solution or key.
Yeah, so there's this biological relevance to this gate, but I think this again shows the power of collaboration here in that just coming from what we'd been working on recently with elastic knots and kind of related to this picture I showed at the beginning of these little loops, writhe loops that they form. This figure of 8 thing is very tantalizing from a topological point of view. It's almost as if what the worm is doing is sort of putting in clockwise tangling and then putting in anti-clockwise tangling in a way that might undo the previous tangling. And that's basically the intuition. And in these types of problems, I think once you have that like picture, you can just run with it. And that's what we were able to do next. So again, I think I won't say too much about this, but there's a trajectory model that we can write down that's a stochastic model for these worms. But then we have to come back to, so that's progress on the theoretical side. And then at that point, we throw it back to Saad and Harry and ask, can we get enough data to verify that there's this trajectory model is real or reasonable? So now we're kind of closing in on something that might be actually functional.
This is where Vishal tells us that I need this head gates, but multiple worms and or many videos, like close to 50,000 or like 80,000 points. This is where I get a revolt in the lab where PhD student at some point says, Saad, I'm not going to do this. But anyways, I bribed him and convinced him that if he did this, maybe there was a science paper. Turns out he wasn't motivated by a science paper. Any case, long story short, a couple of people manually tracked both the left-hand side tangling videos and the high speed. As you know, if you do it at like thousands of frames per second, you get thousands of frames per second data to analyze. And we tracked each single head of every worm, multiple videos. Took us months to get that because there was no automated way to do it. So it was just pure brute force to get Vishal the data. But it's totally worth it.
I like to think that I'd built up some credibility with the time investment that went into producing these videos to be able to ask for something like that in the first place.
We might have to skip the whole phase thing.
Yeah. So, in the interest of time, I think we'll also skip the next bit where we kind of find this mathematical picture. But I'll say that the one of the takeaways here in terms of what the mathematical picture is that what the worms do, it turns out, is very similar to a classic topology puzzle that is suitable for all ages. So this question asks, imagine you have a picture frame and you want to hang it on 2 pegs. So there's 2 purple discs of the pegs. So you're trying to hang this picture frame on 2 pegs such that it falls if either peg is removed. So this doesn't work because if I remove one of the pegs, it stays hung up on the other peg. So this is a beautiful puzzle. And if you don't know the answer and don't want to know, then look away now, because I'm going to spoil it forever. But it turns out the answer is to do this thing where you wind around one peg one way, then wind around the other peg, and then reverse the winding around each peg, which is almost exactly what the worms are doing in this clockwise gate. And it turns out that these trajectory equations that we wrote down reproduce this type of behavior very closely. So the worms are using the solution to this classical topology puzzle to build these tangles that they can unravel quickly.
I wanted to share an anecdote. I prioritize and prize myself and my lab on being naturalists. We're out in the field from Borneo to the Madagascar, Peruvian Amazon. This is where I think this collaboration showed me how even a mathematician can make an observation. And I love this, the armchair naturalist. I didn't have a better word for it, but this is what I'm calling Vishal as a mathematician's in silico naturalist observation. So he comes to us, we invite him to the lab at Georgia Tech, and he hangs out and he says, Saad, and then he told us of some observation he had made in his simulations, and I'll let you describe it. But this small anecdote has sparked off another last 3 years of collaboration. And I find it remarkable. And I think this is another value of a kind of Batman collaboration where we respect each other's ability to drive a question. And we've, as experimentalists, invested a huge amount of resources, defined resources, and I'll show you, based on just Vishal's observation. And we were like, oh my gosh, he's observed something ridiculously cool. So I'll let you describe what he told us maybe 3 years ago.
So there's this... So we talked a little bit about this untangling business. one way to think about it is that when you start with a tangle like this, what we learn from the worms is that there's a special type of helical wave thing that they do that untangles this tangle, dissolves it. So one of the questions that raises is, what if you have a static tangle and you just try and do that with one filament? Can you get a similar type of behavior? And that's what this is. Oh. Sorry for jumping the gun there. That's what the left-hand video is trying to show. So imagine you take one filament stuck in a tangle and you shake it. Can you do something similar to what's happening on the right-hand picture, but from the perspective of one filament? Can you excite these helical waves and get the thing to extract quickly? And does it depend on the frequency? So I think I mean, this question is not sort of an isolated theoretical observation. This process involves a lot of back and forth between experiments and simulation. So on some level, I think what these simulations are numerical experiments that we do alongside the actual experiments in order to build intuition. And none of this is possible without that back and forth process.
So what he said and what I heard is, how do you extract a filament from a tangle? And I called Ishant, the postdoc in my lab, and I was like, oh my gosh, this is a great puzzle. We should build and solve this, because I think what Vishal is telling us is there might be some specific frequency at which you can quickly extract a filament. So this took a while to build, but Ishant basically first we had to create control tangles. These are cotton fibers. And then we had to figure out a shaker. And now Ishant basically is trying to extract one filament out of this tangle, the stochastic tangle. And kind of testament to Vishal's kind of intuition and Ishant's kind of work at it, we've kind of discovered something which we didn't think was possible, is this idea of a topological resonance and this amorphous tangle. And I think this is just beautiful. Essentially what this picture is saying is, this is a story you're developing, that if you wanted to remove, say, one Ethernet cable from a jumble of Ethernet cables in your drawer, if you shake it at the one right frequency, it comes out, and that's the Goldilocks frequency. If you do it too slow, it doesn't. If you do it too fast, it doesn't. Right frequency, you can extract a filament with ease. Does that make sense? Did you want to augment that?
I think that was,
okay.
Yeah.
And with this picture, we went back to biology, and I said to Harry, something has to eat these worms in a blob, just like we have to eat ramen and noodles as I'm eating noodles. And I was like, yeah, we do shake it. So Harry figured out that actually there are freshwater eels that feed on these worms. So he then set about doing predatory experiments. And he discovered what Vishal had observed in this in silico is actually true for biology. Here you'll see an eel predating and yanking at a specific frequency one worm in a blob to kind of extract it so it could eat it and suck, because eels are suckers and they want to suck out the hemolymph from the worms. So I thought this was cool. We wrote this paper on the biology and we're writing the physics one. So again, this kind of math inspires kind of biological observations.
And just to say a bit more outlook, another thing that we're trying to do at the moment is explore some of the space of tangling and tangling structures more systematically. So now that we have a picture of how some of this topological stuff behaves, we can ask questions about more specific scenarios. And there's two types of reductions that are interesting, that immediately jump out. One is, imagine you had a roughly one-dimensional tangle where everything's kind of aligned, and that's this case on the left that we call the worm buoys. Again, these are amazing experiments done by Harry Toison, who has sadly left the lab, but has left
PhD,
us with.
He says sadly, but he left with a PhD, so happily left.
Happily, yes. But he's left us with so much data that I think it'll take several more PhDs to get through that all. But there's a question of how to understand the 1D organization of this. And then there's relatedly, you can ask what happens if your tangle mostly happens on a plane. And then you can ask questions about the 2D organization of this. And this is kind of exciting for me because I started this project, this collaboration, many years ago as a grad student. Now I'm a PI and I have a postdoc working on this as well. So that's Jongshin, you at the top. So this collaboration has really spanned many career stages for me.
So, this is kind of a nice summary of what we've done. We've thought about these topological active matter. Recently, we've also published on these robotic systems. Long story short, we think we've opened a can of worms, and it's kind of a gift that keeps on giving. So how do you replicate this in your own life if you wish? So we jotted down a few things that work for us that we thought would be worthwhile kind of sharing.
Can I jump in quickly
Of
there as well?
course.
So this is something that we only did because we were doing this joint talk, and I think it was actually very valuable to do. So it's not something that I ever thought of before, sort of why this worked, but it was a very helpful exercise.
I think in our case, and most of the collaborations, I think starting with a clear puzzle that we could kind of reach across the aisle was helpful, a weird observation, a puzzle. And I think I was very inspired by Anne. I think she kind of hit the right tone of getting us excited by questions rather than answers. I've come to realize that scientists, we get excited more about questions than answers, which is why sometimes reading a paper is not as much fun as going to a talk, like, for example, Anne, and basically get to the open question. We don't want answers, we want questions. So that was helpful. To kind of get us all both on the same page. And I think the hit rate of 1 on 8 cold emails suggested that the right person was looking at it and said, yep, this is the next puzzle to go. I think we kind of hopefully comes across in the way we're not just talking about it, but also showing you is that we clearly respect our domain expertise and recognize we complement each other and neither comes first or last. We kind of step by step move along as a Batman and Batman approach. And we kind of live this every week. Did you want to comment on this?
Point 3 or
yeah.
So in terms of this Batman and Batman approach, there's this process of learning to speak the same language, which I think from the mathematical standpoint means being able to visualize things and explain things in this visual way that then you can get questions that inform your mathematical picture as well. Yeah. So that was super helpful. And likewise, when Harry was showing us these worm videos, he was presenting them to me in a way that almost felt mathematical, and that I knew that he was trying to get at stuff that would help me from the math side too.
I think this was quite important because both of us are idealists, but I think there is a small amount of pragmatists in us that recognizes that beyond the big idea, we still need to put in the work. So we would have all these meetings, and many a times there wouldn't be much to show, but we would kind of ensure we left off like a baton on each other, some homework, and we would kind of keep this going for many, many years. And I think that was helpful to maintain that language and continuity.
Yeah, and I think Saad's been very kind about this whole equal ownership business. It is true that experiments are much more costly just from a money standpoint. So I think it's important to show that there's a serious time investment happening on the theory side as well here.
As we were talking, we realized something that actually happened, and this is now common in synthetic biology, where you have kind of this top-down approach, the classic view that you can learn a lot by watching. And that's what we were doing with the worms. But I think what really made the story shine is Vishal was doing in parallel bottom-up approach. He was basically taking the Feynman approach that what I can't build, I don't understand. And he was asking what are the minimal ingredients and playing with this. What that started to help is that we were developing kind of this mesoscale intuition coming from these opposite spectra and kind of finding our middle ground. And it was sharpening him in refining our biological observations and vice versa. So I think that was really, really helpful. I think one of the things I've learned as I grow older is that when you have these long relationships, we're kind of very respectful because in this, I think Vishal hinted, he went from a PhD student in Boston to now a faculty at UCSD with married and a kid. Harry, when we had the pandemic. And so one of the things we realized there were many, many times we're sharing this kind of positive story, but there's lots of things that happen. Life happens. And we were, okay to acknowledge that people would be traveling. And Harry is a Navy reservist. There would be time he would be off in a submarine somewhere. And we would just be patient and then pick up the baton again. And, it just, in these relationships, I think the moral of the story here is that our eye was on the long game here and not just one paper. Hopefully, as you see, we have multiple things cooking and we enjoyed the process rather than to kind of one end game here.
One remark I'll make here is that, like Saad was saying, when we started this, I was a grad student, and now I'm a PI, and I have postdocs and students of my own to mentor. And I think this process of thinking about all of these points is actually super helpful, just from a learning how to mentor standpoint as well. because at this stage I can look back and say what were the main sort of what were the helpful things that the PIs did during this process, and how can I replicate them now that I'm at that stage as well. Yeah, so I think this question of speaking the same language, for example, is something that you can really push as a PI to make sure that the students are talking to each other in a way that is mutually beneficial, and to catch any of the sort of small hiccups that can arise along the way. For example, in terms of in delays or keeping the direction consistent and coherent between both sets of people too.
The last thing, probably controversial, but at least it was helpful for us to align our expectations. We both had the same North Star. I had written in Science, he had published a paper in Science, and we kind of had decided together that this was an interdisciplinary story, and that was the audience, that was the format, that's what we wanted to go for. Of course, we knew that was not a guarantee, but it helped shape our expectations. I've had collaborations with roboticists where for them publishing in an IEEE journal or just publishing in a master's thesis or just isolated single author in biology was enough. And so those are kind of things that may not serve to do this math bio thing, but in our case, we were lucky that we were aligned and knew what audience we would go for and how, and it set kind of the game that we were going to play and how we could take on this challenge and avoid this figure 6 kind of paradigm that we wanted to break for ourselves. So I think it helped.
Yeah, you have to want the other person to win. Yeah.
Go and win the way we kind of mutually decided that was what winning satisfactory one would look like for us. Yeah. Thank you so much for paying attention. We're happy to take questions.
I think any audience member who's been through rain with long hair would love to know what is the frequency that you get in both elements out of a clump. So you need to shake.
Are you volunteering?
Are you suggesting a?
So,
you go first.
So hair with wet hair?
Yeah.
One single hair or multiple hairs? Because we have two projects. We have something called fairy knots, which is a thing, which is hair knots, and it's called fairy knots colloquially. So we are studying that problem. But we are also studying the hair tangle problem. Which answer do you want?
I love how seriously you're taking my question. Having a clump of hair into single hairs again. Yeah.
Yeah.
I can think of at least two approaches. There's the untangling stuff that Saad mentioned that Ishant was doing, where you shake the string at the right frequency. There's another thing that we've started thinking about that we didn't say too much about here on braids, which is a classical thing that you will get in hair. So if you have a tangle where all the filaments are kind of aligned, there's a 1D structure to it. Turns out that makes it much simpler. Maybe not from the actual hair combing perspective, but from the mathematical perspective. Yeah, so all that to say, I don't know how to do it yet, but I think it's mathematically nice.
You know what is worse than that is my wife will always give me her necklace, and the hair and the necklace both form a knot. Have you ever had that? Like, that's a beast to solve. Like hair, maybe hair and necklace, two materials.
And the necklace is very interesting because it's not doesn't have normal bending stiffness.
Yeah,
we could talk about this.
Yeah, we will
for days. Yeah.
And
the thing with the, if I may one more, your thing with the eels really fascinates me. So the way eel does it seem like it finds a single tail and kind of yanks it out, or does it sort of randomly attack any part of this cluster? I'm just wondering, how did it detect where to go?
I see. From our data, it suggests that it's trying to observe because it's sensing some of the fluidic motion, so the one that is the most active and grab at it, but it seems random from our data. We haven't resolved it. It's a little bit harder to quantify exactly what it is, but yeah, that's the working answer.
I think we have time for one more question.
Oh, okay.
It's fast. Are the worms in nature genetically related to each other? Are they clonal or are they heterogeneous?
Right. So in nature, worms will sexually reproduce. In lab, nobody has ever been able to do them sexually reproduce. So the way we do it is essentially chop it up. We think of these as wolverines. So you chop them up and they just regrow. So you just kind of mash them a little bit and then leave them, and suddenly all the parts will grow heads or tails, whatever it is.
So it's a cooperative behavior, this clumping among genetically related?
In lab, yeah.
Yeah, okay.
There seems to be one last question, Maria. Do we have permission to take it? Okay, so you're the closer.
right? Like types of environment, like physical properties. So thinking of there's other big field of parasitology is the helmet. So how this would be, and I know that they also form clumps like this inside the body.
Yeah.
So are you interested in knowing like what happens in that sense when they're exposed to everything that we bring?
Very interested. In fact, I tried to write a grant to Gates Foundation. It's called, I forget what it is, but you know how cats or something will eat a mothball or a hairball? I thought it was a great way to think of a drug delivery problem because, you could basically give a worm ball and then it would deliver drugs. In my head, it made lots of sense. But yeah, I'm very interested in thinking about mothballs and hairballs and how they might get tangled up in a system and how we might use it to deliver drugs or something like that and then untangle in this bits of the system. But yeah, helminth sounds great and other parasitic worms sounds fun. Thank you so much again. Thank you.